Well, hello everyone, and welcome to our latest blog, The Golden Age of Genes, from DNA to Donated Hearts. My name is Graham Swinard, and I'd like to introduce my friend John Bryant. We're co-authors of a recent book on science and religious faith from the perspective of a physicist, that's me, and a biologist, that's John. The cover is, as you can see, displayed on a backdrop, and you might like to visit our website. We've got the address up there on the screen now. Now, today's video blog focuses on John's expertise, biology, and in particular, what's going on in the world of genetics. So, John, you are on record as saying that genetics is enjoying a golden age. Why do you say that?
Well, I still find it amazing that it was only at the end of the Second World War that we showed that genes were made of DNA. We knew that DNA existed in chromosomes, but nobody was sure that it was the DNA part of chromosomes that contained the genes, but it turned out they did. And of course, that led to a great flurry of research. And while I was a little lad at primary school, Watson and Crick discovered the double helical structure of DNA. And that structure gave quite strong indications of how the DNA actually works as a piece of genetic material. That was really quite nice, that you could see from the structure how it might work. And that just basically led to an explosion of research. And so by the time I was a student, the genetic code was being decoded, and the mechanisms for actually turning that decoded code into working molecules in the cell were being worked out. Lots of that was happening actually in Cambridge, just down the road from where we were having our lectures, so it was quite an exciting time. And so I suppose that period then set the scene for our current understanding, that we showed, for example, that bacteria could move genes between themselves, and so we could use that for genetic engineering. And so it went on and on, methods for sequencing DNA, etc., etc. So DNA became much more a central part of biological research, and understanding how it worked became a central part of our understanding of how living things actually exist.
So for you, John, what were the real highlights?
Yeah, that's quite tricky, really. There's so many of them, but I think what I like in kind of general terms is the way that one discovery or a group of discovery leads on to others. And so we can use a group of discoveries to develop a technique, and that technique in turn will lead on to further new findings. Genetic engineering is a classic example of that it's based on techniques which were discovered by research. We can use it in useful applications, but it's also got a vast array of applications in research on genes. And so in my lab, for example, we use genetic engineering techniques to look at some of the ways in which genes are switched on and off. Another example is understanding how DNA is copied in cells, how a cell gets ready for cell division by copying its DNA. That led on to the ability to sequence DNA, that is, to discover the order of the building blocks. And that gives us a huge range of applications, which we might come back to later.
I can understand how the relationship between research findings and research techniques is a kind of virtuous circle. But do you have any favorite research findings?
Well, again, that's tricky. I suppose it's a bit self-centered to say one of my favorites was discovered in my own research in collaboration with one of the government-funded labs, when we discovered the way that a particular virus copies its genome, replicates its DNA. It was totally unexpected, this method, this technique, and it's been a kind of core finding for our understanding of that group of viruses. So that's one of my own. But I think another general finding, which is quite surprising, and it's a bit quirky, is the way that plants can communicate with each other to switch on or off each other's genes.
Wow.
And that's,
how does that work?
well, it can work in at least two ways that we know of. So, for example, if a plant is invaded by a pathogen, immediately there's cellular damage where the pathogen invades, whether it's a bacterium or a virus. And as David Attenborough pointed out on Green Planet, the cells in that plant can communicate with other cells in the plant to say, hey guys, there's danger at hand, and prepare defenses. But more than that, the plant can synthesize molecules which are airborne. We call those volatile molecules. And other plants that are prone to being attacked by that pathogen can get their defenses ready before the pathogen arrives. So they are quite literally communicating with each other via volatile molecules, a bit like scenting a perfume and knowing there's perhaps another person coming by, maybe of the opposite sex, but that's not true of the plants here, of course. And another way they do it is that if you go out into the forest, many of the trees are linked by a huge network of fungal threads, which we call a mycelium, and we talked about that in the book. And they can communicate gene control molecules along those fungal threads. So
I found that amazing, actually. I did find that amazing.
Yeah, it is. It is.
Anyway, earlier you said that you wanted to look at DNA sequencing in a little bit more detail.
Yeah. Yeah, DNA sequencing. Well, actually, I'd like to come back a little bit more to human genes before we do that, because the DNA sequencing, which I'll come back to, has enabled us to know that humans, in common with all mammals, well, probably all mammals, have got many fewer genes than we have genetic traits. And so how does that happen? It means that some genes must be multifunctional. And working out how the cell decides which of the functions to use is a really major challenge. Plants have some multifunctional genes. We discovered one of them actually, but it's not such a big thing in plants. And so that's a sort of a challenge that's arisen from DNA sequencing, which is giving us a light in genetic control mechanisms, if you like. But DNA sequencing itself, I think it's important because understanding the sequence of genes, as I've just hinted in talking about multifunctional genes, understanding the sequence of a gene helps us to understand its function. It helps us to understand where it might go wrong, or how it's controlled, and how its control mechanisms might go wrong. And so you might get genetic disease. That's a disease that's inherited through genetics, or you might have a disease that's caused by a malfunctioning of a gene within the body, like cancer, but it may not be inherited. So understanding DNA sequencing is great in science, but it's also great in medicine. We can use it in diagnosis in a very specific way, which we can think about later. If you can share the screen and have a look at this slide here, we can see some of the progress that's been made here. So yeah, look at that. So this is about how much it costs to sequence a genome. So over on the left, you've got the approximate date of finish of the Human Genome Project. It was actually very slightly later than that. But when the Human Genome Project was initiated 10, 11, 12 years earlier than that, it was reckoned that to sequence an average human genome cost $3 billion. And by average human genome, that's how the Human Genome Project worked, that bits of human DNA was sequenced all over the world, and the data were all collated in 3 centers. And finally, the overall collation was done in the States. But by the end of the project, because of improved technology, the cost of sequencing had come down to a mere $100,000, cheap compared with $3 billion. But you can see what happened in 2007 is that two different techniques were invented for sequencing DNA. And they're both cheaper and they're both much more rapid. And so by the time we've got to last year, we're down under $1,000 for sequencing a human genome. And now I don't mean an average genome, I mean my genome or your genome, Graham. We can sequence the genome of an individual human for $1,000. And that's had a huge amount of spin-offs in medicine, but also in science in general. We can look at the genome of any organism and see how the genes maybe have changed in evolution from closely related organisms, etc., etc. It's really been a very, very huge advance in progress.
Yeah. Okay, let's see if I can get back. Oh no, I think you want to move on to the next slide, actually.
Yeah. I mean, if you look, it's not just cost, but these two new techniques. One of them is particularly fast. It's these new techniques that have enabled us to speed up the process. As I say, 10, 11, 12 years to get an average sequence for the human species, and now we've got the ability to look at one individual genome. Incredibly useful when we're trying to study whether a family's got a gene running in it that's difficult. And one of the techniques would allow us to do it in maybe 24 hours, maybe a bit longer. The more rapid, not quite so accurate, but sometimes the high degree of accuracy is not quite so important. We'll do it not in just 24 or 36 hours, but in rather less than that. And so I didn't know that the Guinness Book of Records has a genome sequencing section, which is really quite extraordinary. But if we look at that headline, the headline referred to a human genome sequence in under 8 hours, 7 hours something. Quite absolutely extraordinary.
Yeah.
And the spin-offs that's having, particularly in medicine, a diagnosis using that technique enabled a diagnosis of a boy with a particular condition that they didn't know whether he had it or not, and the diagnosis was achieved through very rapid sequencing of his genome, and it went into the Guinness Book of Records, which I think is rather amusing.
Wow. Yeah. Is there anything more that you'd like to say, John?
Yeah, I think just one thing, going back to that the virtuous circle we talked about between discoveries leading on to techniques that lead on to discoveries. And I think a relatively new technique called genome editing fits that bill perfectly. That we now understand when viruses invade bacteria, and it's a special sort of virus that does that, bacteria can defend themselves by targeting in on particular bits of the virus DNA and snipping it, breaking it, and sometimes removing a section. And we call that genome editing. And it really is like, those of us who are old enough to remember, it really is like editing a cassette tape or a film. You cut a piece out and you join the cut ends. And genome editing does exactly that. And it has a huge range of techniques, again, in understanding how genes work and so on, and maybe improving crops by removing genes that hinder growth, etc., etc. But one particular application, which I wrote about in this book, I must have a quick puff of this book. Introduction to Bioethics, second edition. One particular application was to use genome editing to snip out from the pig genome those sequences that make the pig organs unsuitable for transplant into humans. The organs are the right sort of size, and pigs are metabolically similar to ourselves. And transplanting organs from pigs has long been thought of as a way of dealing with the shortage of organs. It's actually happened that the proof of concept was achieved by a human heart, a pig heart being transplanted actually into a human from a pig that had its genome edited in the way that I just described. And here it happened in America. The nasal oxygen tube is a little strange, but here is the happy patient who's now got a pig heart working happily within him, and that happened last month, and so we know he's still alive 3 weeks later. So understanding genome editing led to an application in medicine, and that's something we see, right across the board with modern genetics, applications in medicine and research, and in agriculture. So long may it continue. Long may the golden age continue.
Yeah, absolutely. Yeah. So that's absolutely amazing, John. So thank you for all that. There's a very illuminating glimpse at what's going on in the world of genetics, and indeed an insight into developments by your own team and in your own laboratory down there at Exeter University.
Yeah.
I have to say that I have no expertise in biology, but since our liaison in leading conferences together and writing the book, I now find myself reading popular books by the biology of life, which I find absolutely awesome. And I suppose it could be said that the psalmist once found the glory of God in the majesty of the night sky, but now, thanks to the work of the life scientists, we can see his glory within us. So that's amazing. So anyway, so thank you for listening, everybody. Whatever you're doing today, have a good day, and farewell.
Bye.
Bye.